Hello, we're glad you've joined us for this live webinar, Automation in Core Laboratory, Experience of a Large European Academic Hospital. I am Christina Mahalik of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars, advancing scientific collaboration and learning. It's brought to you by LabRoots. To learn more, visit labroots.com. Let's get started with today's presentation. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will be open when you click on the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems hearing the presentation, just click on the support button found at the top right of the presentation window, or report the problem by clicking on the green Q&A button at the lower left. Additionally, this is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your web page and follow the process of obtaining your credits. I now present today's speaker, Professor Damien Gruson from St. Luc University Hospital, Brussels, Belgium. Professor Damien Gruson was awarded his degree of pharmacist and later of specialist in laboratory medicine from the Catholic University of Louvain, Brussels, Belgium. He joined the Department of Laboratory Medicine of the St. Luc University Hospital in 2008. He is now Associated Laboratory Director and leading endocrine biology. He is also a member of the Research Unit on Endocrinology, Diabetes, and Nutrition of the Catholic University of Louvain. Professor Damien Gruson has published numerous articles in several international peer-reviewed journals. Professor Gruson is a member of the Committee on Distance Learning for the International Federation of Clinical Chemistry and Laboratory Medicine, IFCC, of the IFCC Task Force for Young Scientists, chair between 2010 and 2012, and now consultant. A member of the Division of Endocrinology of the American Association of Clinical Chemistry. Professor Damien Gruson is also a fellow of the European Society of Cardiology. Professor Gruson will now begin his presentation. Professor?
Yes, thank you very much for this very kind introduction. I'm delighted to be with you today. So good morning, everyone, and discuss about our feedback about automation in a core laboratory of an academic hospital. This academic hospital is the Clinique Universitaire Saint-Luc. We are located in Brussels, so the capital of Belgium. The hospital is around 1,000 beds. Is part of the Catholic University of Louvain. And we are lucky to have new lab facilities since 2005, which is the Tour Rosalind Franklin building that you can see here. The hospital is just there. And in the surrounding, we also have the research unit here from the Catholic University. The laboratory characteristics are the following. We are processing around 12 million analyses per year. In the lab, we have 250 employees from different competencies, lab technologists, administrative managers, pharmacists, and MDs. And I will show you later the detailed organization of the lab building. We have a core laboratory on the ground floor, processing around 60% of our activities, and these core laboratories was submitted to the automation process in 2013. We went live in June 2014, and so I will present you today the experience around this automation project. So when you start automation project, you have some ideas in the back mind. We know that automation will define technique, method, or systems of operating or controlling a process by highly automatic means, electronic device, in order to reduce human intervention to a minimum. And what we are expecting in all fields is clearly more automation to contribute to more patient safety and more safety of lab workers. What we know also is the dogma published by in a famous report of the European Commission: to err is human, to delay is deadly. So automation should allow us to reduce errors in the different phases of the analytical process, but we have also the opportunity to rebuild the lab process and speed up the delivery of results to the physician. So improvement in the turnaround time of analysis by better design of your automation lines, automation process, and the related workforces. When you start an automation project, you are coming with a lot of hopes for a lot of people around you, for your physician. They will be stimulated to learn that the turnaround time of analysis can be improved, that the menu of analysis available and automated can be broader than before, with an improved frequency of testing for some assays. The patients will really be responsive to the keywords around patient safety, improvement of laboratory outcomes. So it will be up for them also. Lab technologists will be, I will say, afraid at the beginning because it's a major change for them. So you need to involve them as soon as possible in the beginning of the project to get the buy-in, but they will be stimulated also because they know that the automation of manual tasks in the pre-analytic phase can allow them to spend more time to what is critical: review of results, monitoring of equipment, validation. Us, lab managers, medical directors, because we have the opportunity of new equipment, improved assays, improved process, clear perspective to redesign your lab facilities, and of course our best friends from the management, both the hospital or the lab managers, where automation means also economy of scales and also redistribution of the value on specialty tests and related incomes. So ops is something related to automation project. Fear also, clearly, because we know that it's a major challenge for everyone. A lot of work, a lot of preparation, a lot of validation, so major impact, and we need to be prepared for that. And we need to bring all the competencies in the team to get real teamwork and again the buy-in around the project. So don't be don't be afraid, just be prepared and know that the road will be long and difficult. Several steps will be considered, I assume, for the automation. The first focus will be on the pre-analytics and the pre-analytical phases. As everyone knows, the analytical process is a cycle. The cycle, according to the ISO or CAP accreditation guidelines, should be carefully monitored and controlled. And we have three major phases: the pre-analytical phase, the analytical phase, and the post-analytical phase. And we know that manual tasks remain in some of these phases, which could be related to increased number of errors. According to Marius Plebani's numbers, clearly most of the error occurs for the pre-analytical phase and could be related to handling problem, ID on the tubes, which will be more pre-analytical matters, the selection of the appropriate tubes or matrix for the analysis, and there is also all related to the lab pre-analytics, insufficient sample volumes, interferences, decaping, labeling. So all these source of errors lead to this 46 to 60% of errors in the pre-analytical phase. We see that the numbers are lower in the analytics because they are already more automated, 7 to 13% of the errors in the lab, and we have the post-analytical rate 19 to 47% related to inadequate turnaround time, incorrect data entry or reporting of analysis, which also could be influenced by the pre-analytics. So the work on the automation of the pre-analytical phase will clearly impact the whole analytical cycle. So it's a specific focus for the automation, and fortunately that's where we have a lot of solutions from the in vitro diagnostics products and companies. So we know, and we know where are the errors. We can admit that, and we can work to reduce the number. So what will be really important in the preparation phase of the automation project that you could have is the mapping of the lab activities. And the mapping of the lab activities, the cartography, will identify the steps remaining manual in the pre-analytics, and you will find on the market solution to transfer automatically the tubes, to transport them, to decap them, to sort them, to aliquot them. So you will reduce these manual steps: centrifugation, decapping, transfer, aliquotings. You will clearly simplify them, reduce the related risk of errors, and improve the safety of the workers on these low-value manual tasks. So clearly, do the mapping, monitor the number of steps that you decrease in this pre-analytics, and you will decrease the risk. You will improve the turnaround time. That's clear. So mapping, cartography, and targets based on your cartography. The impact on pre-analytical phase is described already in the literatures, and we have data from several groups, which could be the one from Salt Lakes and Charlie Hawker's group showing that when you are automating your process, you are decreasing the number of lost specimens according to the different steps of automation. So more visibility and traceability on the samples load on your automation track, decrease of the monitoring, improve for the errors. So mislabeled tubes, inappropriately collected tubes are maybe more easily tracked when you have the automation system on. What we observed in Brussels is the following. Here are the data from the central labs, and you see in green the percentage of the time of our technologies spent on analytics or validation, the technical validation. After the automation process, you can see that we clearly improve the percentage devoted to the green time. So decrease the time spent on pre-analytics, increase the time and attention spent to the analytics and control of the process and sample quality, which is really important. So that's for the central labs, I would say the routine test. What did we observe in the specialty testing process? So the impact of the automation on specialty labs was also the same conclusion by being more efficient to prepare the tubes for these specialty teams. We decreased the time spent to tubes preparation, sorting, and clearly again improve the green area, which is this time spent for analysis and the time spent for review of the quality of samples and results. So decreasing the loss of time related to unanalytical action. So reconcentrating the workforces on what is important for the technologies where they can put values is the analytical phases. The effect of automation is also anticipated on the analytical phases, and there is clearly a need. I can just take a simple example, the one of the troponin testing. We know that we have since few years ultra-sensitive troponin assays. These ultra-sensitive assays give us clearly better analytical performances at the low concentration and around the 99 percentile, which is also improving the accuracy of measurement, the ability to detect lower concentration, and the ability to speed up the triage process in some target services like the emergency department. And that's why the recommendation of the European Society of Cardiology have decreased the time for triage from 12 hours to 6 and 3 hours. There is this question around the potential of 1 hour route rule-in, which is perspectives. But clearly, if we are not able to stabilize and improve the process from an operation perspective, we will not be able to fit to the guidelines around this reduced algorithm for triage. So there is clearly, from the revision of the process and the automation, the capacity to provide test results more quickly and fit with guidelines. That has been described also by several groups. In Singapore, we had impact on the mean turnaround time of analysis for the routine and for the stat samples, so pre and post automation. And it's also interesting to observe that there is a decrease of what is also a very good indicator, the 99 percentile. If you are decreasing the 99 percentile, or if you are able to more control the 99 percentile, you reduce the number of outliers, you reduce the number of complaints, you have a clearly more stable process. Same observation from the French group of Alain Donizeau in Lens. By automating steps, you are decreasing for the chemistry the turnaround time really significantly. So decreasing the time of response and better control of the whole process and more stability for the report of data. And that will be something that you will find abundantly in the literature, control of the mean, control of the median, but clearly decrease of the number of outlier percentage in the lab. So if you are decreasing the number of outlier, again the satisfaction from the physician or the clients will be better. Again, some data from Italy on the left, showing the number of outliers pre and post automation, and again the benefits of automation. What is interesting to observe also is that if you are automated the operation in the central lab, you will have the control of a better control of the turnaround time. If you have a better control of the turnaround time, the results will go fast to the physician, who will reduce the number of additional requests. So what can happen is, if the physician do not get the result on time, they think that you are not processing the tubes, and they will again order the same analysis. So by controlling the process, by controlling the speed and the frequency of reports, you can impact the lab test prescription, decrease that, and get some other benefits. So again, example with number, you see on different parameter of chemistry that automation is impacting the delivery. Troponin 99 percentile were reduced from 74 minutes to 57 minutes. The creatinine, the glucose, the potassium, same effect. So clearly the ability to get this efficient monitoring and release of results. Impact of the hospital, you can also decrease the length of stay. What we observe here in Brussels is that we are clearly better response after the automation to the need of the hospital, which is the patient out of the hospital around 11 a.m. after an hospital stay. And we are clearly now more efficient by shorter turnaround time in the morning, more adequation with this hospital request. And I will have some example of turnaround time in few slides. What we also did is, by the connection of the analyzers on the automated track, we clearly improved the availability of some assays. So broader menu, better responding to the physician needs. The scope of our automation project was involving the chemistry testing, the endocrinology testing, serology, virology, tumor markers, and hematology. And we have this broader menu on board which is good for the inpatient, but which is also good for improved response for the patient from the outpatient clinics. We automated 17 assays for therapeutic drug monitoring, 36 assays from virology, specific protein and tumor marker, 43 assays, 6 panel of assays for hematology, 42 tests in endocrinology, 75 in biochemistry, 35 in autoimmunity. So hundreds of tests were involved, were, I will say, improved, but there is the counterpart, which is an tremendous work to re-evaluate, validate the method when they were changed, and also redefine some reference values when the assays change from method or manufacturer. So it was clearly a huge work of for us, and we are still closing some validation sheets to be consistent with the accreditation process. Okay, anyway, you can see the benefits in terms of turnaround time. So previously we had for the stat samples in immunoanalysis, pregnancy test, troponin I, we had a median of 1 hour 20 minutes. The percentile 90 was 1 hour 42 minutes. And after the automation for the chemistry and stat immunoanalysis samples, you see a fall of the median, only 37 minutes, which is really excellent for release of troponin results. And P90 was 51 minutes, so we significantly improve both median and P90. We clearly stabilize for the routine immunoanalogy testing, immunology testing, the median and the P90 also. You see outliers were there, and now 142 hours for the median, 253 for the P90, which means that patient coming in early morning or early afternoon for consultation of endocrinology, thyroid, virology will get the result early in the morning or early in the afternoon. So clearly improvement for everyone. Hematology, by the automation of the hematology process, we improve the medians, and we also improve and stabilize the whole process by decreasing the P90. Anyway, the message that I will give you also is that there is not one solution for all the lab, and we need to customize what we want. And clearly, there is a work to do with the manufacturers involved. As one size does not fit all, you need to, in the project management phase, confirm the needs, work on the needs with the partners of the project, heavily companies, define what you want, and try to implement what is the best for your labs. I'm back to the structure of our laboratory. So I present you the view of the building at the beginning. Our specificity is the following. We have the core lab here at the ground floor. And what's happened is that we have our specialty labs at the upper floors, and the central pre- and peri-analytic operational process also in the core laboratory. So one of our requirements is, in addition to the routine, we need to take care of the delivery of the samples or aliquots to the specialty testing labs. So there is a clear need from us to get this strong aliquoting process, to get this parallel transfer of primary tubes and aliquots to the targets, both in the routine lab or in the specialty testing labs. That was one of our wishes. So what we did to get that is the definition of, I will say, at least 3 flows. We have one main flows with the automation of the pre-analytics, their centrifugation, decapping, aliquoting for the chemistry, immunoanalysis, urine, coagulation test, virology, serology. So all the epanionized plasma tubes, serum tubes, urine, citrated tubes will go just with this single point, flow number 1. Flow number 2, the red flow for us is the meaning of EDTA tubes, which mean automation track for the hematology, so one line there. And what we did is the sorting of the tubes here, sorting of the tubes, EDTA tubes, mean not only the hematology, but meaning also of the tubes devoted to immunosuppressant testing. Immunosuppressant testing will be performed on an upper floor by mass spectrometry. So it's really important to sort them quickly there to respect the turnaround time of mass spectrometry. And we have a third flow with another automated module here to help, which is the sorting of the aliquots. Aliquots are prepared here massively, but they will be classified by targets, and the targets can be mass spec, the targets can be specialty immunoanalysis, can be radioimmunoassays. They will be sorted by this module in order to reduce the preparation work by the specialty labs. So we have one peri-analytical unit managed by what we call production operators. So they are specialists in the peri-analytical task. So they are preparing, loading the automation lines, and sorting the tubes for specialty labs. And we have the instrumentation related to tracks or here specialty immunoanalysis connected also by the middleware. So we have designed what we called a recursive flow. So the aliquots will be prepared in parallel to the processing of the primary tubes, and aliquots will be sorted by the production operator and then stored at the appropriate temperature, room temperature, fridge, or -20, -80 degrees according to the target and according to the frequency of testing based on the calendar defined by the specialty lab. What's happened because of this recursive flow and implementation of this strong aliquot process? We reduce about 1/3 the number of tubes collected from a patient, so which has an impact on the well-being of the patient. Lower tubes, lower volume of blood collected, and also a cost. By reducing the number of tubes to be collected, we are reducing the cost of the tubes we have to order per year. A key of success for the automation is clearly the IT monitoring and controlling your solution. So the middleware will be a specific focus for you on the automation. We gain from the middleware perspective with one central middleware in the laboratory, core laboratory, controlling the chemistry, immunoanalysis, the hematology, specific protein, allergy testing, the pre-analytic systems, and also some other specific electrophoresis instruments, or endocrinology or virology performed on the liaison. So a clear, common view and common control of the instruments, of the sample traceability of the QC on the middleware, which is clearly simplifying the management by the lab technologist. Middleware is also clearly a way to get improvement of your dashboard and better control of your indicators. You will have more metrics to control the turnaround time of analysis, the number of failures or errors or bad quality samples. You will control the number of reruns or add-on. You can track your reagent consumption and ordering, and clearly added value for validation, technical validation from the lab technologist, monitoring of the QC and the calibration in a more central and common way. So that's benefits from the middleware, and key again in your automation component. It will be also perspective for you to identify the samples that could be rejected because not appropriated for correct results delivery, by identifying more easily the hemolyzed sample, mislabeled or miscollected samples. So improvement is also by the tracking of errors, poor quality samples from the middleware. So globally, automation means middleware means standardization of the controls through IT, and improvement of your indicators, monitoring, and dashboards. And it's like the cockpit of your new A380 instrument. That's where you will control the efficiency of the engine and the checklist allowing to take off every morning by a global view of the QC and calibration. So dealing with automation means that you will have also to consider your business plan before automation. So the finance and the benefits, the economy that you can expect from the automation, and considering all the report in the literature, automation is always related to an improvement of productivity. So you are increasing the number of analyses or build units performed per full equivalent employee. So you are improving the productivity. What you can expect, and what we did, is to send the human resources that we gain from the automation to specialty testing lab, where we are still needing human competencies. So you have two benefits: productivity, redistribution of the value by more workforces on your specialty test, if you are, of course, dealing with the specialty test issues. So more time for specialty tests, more time for validation of innovative tests. What is really important is the perspective to also gain time to spend on the accreditation, manual tasks of procedures, quality monitoring, education. You can train them more efficiently, so you can clearly get benefits for your employees by more time devoted to the education and formation. We have also clearly the productivity, and it's also a perspective for you to redefine your contract with your manufacturers, and you can renegotiate the price billed per test. So, and there is the effect of consolidation, decreasing the number of units after automation. That's what we had from before the automation, so 18 analyzers to do the perimeter of analysis that I showed you previously, and 8 softwares to do that. And after the automation, we add just 9 analyzers and 4 soft, including 1 more global middleware. So clearly the simplification for the process management and the control of the systems. Economy, as I mentioned, by the decrease of, sorry, it's in French, a decrease of number of tubes collected per day, around 900 tubes per day were economy, which is a meaning of around 30,000 euros per year for us, but also an estimation of around 10,000 or 15,000 euros by a reduction of the crashes for these samples. And the big volume, 1,595 liters of blood of economy per year. To reach this target, you will need, and I'm sure that you know that, to build a strong project team and to communicate with all your stakeholders from the lab, from the central lab, lab technologists, administrations, but also communication with the physician to catch up early what they want. So it's just a question of bridging competencies all together and make them working in the same direction. You will need your lab technologists in, IT guys, your medical directors from different areas, hematology, microbiology, chemistry, technical resources from the hospital, because you can need to adapt your facilities. So at the beginning, please bridge your competencies in the lab. You can require the additional competencies from the institution, technical, financial, but you will need them on board to reach all the targets and to deal with all the aspects of the automation, which are the clinical benefits, the operational benefits, follow the change and implement the change, so change management and training of the team, information and technologies and middleware clearly is a specific point of interest. Work on the scalability of your solution, so anticipate on your future needs, and the cost of a strong business plan to prepare them to negotiate with your management for the acquisition of the equipment, and later on to monitor the cost of your production unit. Communication, change management also with the medical team. So it's clearly important to understand what they want, what are the targets in terms of turnaround time of analysis, the menu of tests that they want in your automation solution, and that could be also customized to your institution. Change, so make group of change with your technologies to anticipate and make them install in the project. So I think that by constructing automation, there is also clearly potential or an opportunity to construct or enhance human interaction in your lab facilities, but also with your partners, physicians from the hospitals or subcontractors from the outside. Do we have more perspective related for the automation? Yes, because there is still some point where efficiency is needed. and there is more and more automated system for the pre-analytic task management, so labeling of the tubes. We have more perspective of automation on the analytics, so more modules for the automation of the urine analysis. Molecular biology is coming to core laboratory and could be connected to tracks, and we have from manufacturer also perspective with new instruments, new automate with mass spec detection, so that clearly more perspective for core laboratories. And the difficulties were related to post-analytics, as sometimes we can not only need refrigerated condition, but -20, -80. There is more and more modules customized to be connected on the tracks and allow you to get also the automation of this post-analytical phase and the more efficient monitoring of the tube. So citation from Ambroster really appropriated to automation project management. Ahmad Victoria Kuram, victories love careful preparation. I think that's all what we need, with a deep analysis of the needs, and then define what you can expect as benefit, monitor them, and interact and communicate with all your partners, team members, to make it a great success. So I would like to acknowledge also Catherine Filet, which is the medical lead of the core laboratory, and I would like to thank you for your attention today.
Well, thank you, Damien, for your presentation. It was very enjoyable. A quick reminder for our audience on how to submit questions. Simply type them in the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Professor Gruson will answer as many questions as time permits. And looks like our first question to you is, how do we provide solutions to the maintenance challenge of automation?
So how do we provide solution to the major challenge? I think that it's mostly in the project management way. So you need to set the project, nominate project leader, and involve the competencies who can help you to manage that. So clearly multidisciplinary needs, project management, communication, consultancy with IVD manufacturers, try to anticipate visits, labs where automation have been done, and trying to learn from them. So that's some keys to help you in such a big project.
Excellent. Thank you so much. As it looks like we're running out of time, if there are no more questions, I'd like to once again thank you, Professor Gruson, for your presentation. Professor, do you have any final comments as we wrap up the presentation?
Much.
Oh, our absolute pleasure. I'd like to thank our audience for joining us today and having your questions.
So thanks again to LabRoots team also.
Oh, excellent. Thank you. Thank you. And we would like to thank our LabRoots for today's educational webcast. This webcast can be viewed on demand through February 2017. LabRoots will alert you via email when it's available for replay. And please, we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye from LabRoots.